So let me just start sharing my screen, and let me just start the first lecture. Okay.
Yeah.
So let me just do a screen mode. Okay. So I'm going to give you a series of lectures about selected topics in the analysis of large dimensional data. And the plan is like this. So we are going to have it split into 4 parts. So in the first part, I'm going to talk about testing for global null hypothesis and about multiple testing. In the second part, I'm going to talk about identifying important predictors in large databases based on information criteria. Then I'm going to talk about regularization techniques in unsupervised learning. And finally, about reduction of dimensionality by PCA and subspace clustering. So this is the general plan. I'm not very good in following these raising hand and so on. So if Isabella, you could have a look. So I will be happy to answer questions during the talk. If anybody has questions, please ask. So like a kind of an outline of a talk was inspired by the lectures of Professor Emmanuel Candes from Stanford University. So if any one of you find it interesting, I suggest you after this series of lectures go to his webpage. He has a very good teaching materials for many things related to the analysis of modern data, to the modern statistics to deal with such data. And I'm also going to add material from my own book and the chapter, which I'm going to give you the references later. So the classical example of dimensionality reduction is the principal component analysis. So let me just look at it a little bit from the mathematical perspective. So let's say that I have a large data matrix with n rows. So we have n records and p features. Let's assume that these are numerical values. And principal component analysis finds the direction which span the data set which explain the largest part of variability of this data. And now if you look at it from a mathematical perspective, a kind of assumption which is behind the principal component analysis is that this data matrix can be represented as the sum of 2 matrices. M is a certain low rank dimensional matrix which represents the signal, and you have a random noise, a matrix which contains random noise. And our goal in many applications of PCA, our goal is recovering M. So we want to recover the low rank matrix which spans the whole data matrix. And we basically can think about it like separating signal from noise. So we want to separate M, we want to denoise this matrix, eliminate the noise. So what is the general purpose? Well, we might try to understand the biological or economical phenomena which generate the data. We might want to get the data compression. So like identifying few basis factors, which few basic directions which span your data, which might contain most of the information. So you do not have to keep the whole data matrix in your memory. You might reduce the data by basically saving only this information which is contained in the principal components. And also, in some cases, we use such a dimensionality reduction to impute missing data. So this is like a general goal of a large data analysis. We want to separate the signal from noise. We want to identify low dimensional structures which span the data which are noisy. And now, what is a major problem in such a data analysis when you have many features which you measure and you try to look for patterns in such a large data set? Well, the point is that if you have such a large data set, then you might see some patterns which might look like biological phenomena, which arise simply due to a random chance. Just because you are looking at many things, they all behave randomly. Sometimes you might see patterns which just result from the random fluctuations and do not represent the real signal behind the data. So we call such a problem a multiple comparison problem. And in some situations, it is multiple testing. So like in case of PCA, you might need to decide what is the rank of this M matrix. So how many principal components you want to keep. And if you have large data, if you have many features, for example, some of the principal components might appear to explain huge part of the variability of your data, which is only due to the random chance. So we need to decide in PCA how many dimensions we want to estimate, or how many non-zero singular values in this SVD decomposition of the M matrix we need to keep. So I'm going to talk about it in the last lecture, actually. So we are going to talk about PCA in the last lecture. And let me just now start from the basic problem, the easiest problem which we can analyze in the setup. So the problem of multiple testing. So like, let's start from the example. So we have X matrix, we have N1 individuals which are healthy, and for each of them we measure expressions of P genes. Measuring expression of a gene is like measuring the amount of messenger RNA which the gene produces. And then we might have cancer patients, and for these cancer patients we also measure the expression of these P genes. What is the assumption which we are going to make in our statistical analysis? Well, we assume that our axis, so that the rows of our we assume that our axes are IID with expected value equal to mu1j and variance equal to sigma square 1j. And also we have for each gene in the cancer patients, this is going to have, the gene is going to have expected value, expression of this gene is going to have expected value equal to mu2j and have some variance which is sigma squared 2j. Okay, so, and the measurements for each individual in the healthy group and the cancer group, they are independent for each gene. And now we want to test the hypothesis that the gene is associated with cancer. So we want to identify if the gene is associated with cancer. So basically we want to find out if the average expression level of a gene for the healthy patients is different from the cancer patients. So what do we do? Well, you probably know the classical t-test. So we test the hypothesis that the mu's are the same. So there is no difference in expected values of our expressions in healthy and cancer patients for the J gene. And we construct the t-statistics. So we basically calculate the average expression in one group, in the other group. We subtract this, and we divide over the estimator of the standard deviation of this difference. And now, if the sample sizes are large enough, then we can approximate the distribution of the t-statistic by the normal distribution. And basically, all we need is that the variances of this x and y's are finite. This works by the central limit theorem. So if your sample sizes are large enough, you can think about this t-statistic as having a normal distribution with some expected value and variance equal to 1. So I reduced my problem to the generic problem about testing the hypothesis about the expected value in the normal distribution with variance equal to 1. Okay, so what are we going to do? Well, we are going to reject the null hypothesis when the absolute value of my xi, so now xi, please remember, this is my t-statistic. So this xi value is going to be large if there is a large difference between the two populations. And from now on, we are going to talk for a while about this generic problem of estimating mu for the normal distribution. So we are going to reject the hypothesis that mu is 0 if the absolute value of x is large. And what happens? Okay, so I'm doing such tests for P genes. P is in thousands. So we currently can measure the expression level of thousands of genes. In some situations, we do multiple testing when we have millions of tests to do at the same time. So in case when all your hypotheses are true, so all mu's are equal to 0, when the maximum of the absolute values of your t-statistic will behave like the square root of 2 log p, where the p is the number of tests. So if you want to separate the noise, so you don't want to identify that something is important when it is really not, you need to make your critical value increasing to increase with p. So when the number of tests, number of genes you test for is increasing, you need to increase this critical value. And now for a very short while, I'm going to talk about the global null hypothesis. So at first, let's consider the situation that we simply want to find out if our data contains some information. So we tend the hypothesis that all mu's are equal to 0. And one of the procedures to do this is the Bonferroni procedure. So we basically look at the maximum of the absolute values of our statistic, and we are going to reject this null hypothesis when this maximum absolute value exceeds the quantile of the normal distribution at the level 1 minus alpha over 2p. And it's very easy to show that the probability of type 1 error of this procedure is controlled at the level below alpha. So alpha is the significance level or the probability that you are going to reject the null hypothesis when it is true. So with a very simple calculation, you see it's simply based on bounding probability of a sum of events by the sum of probabilities. Interestingly, so this seems like pretty much very much redundant, this bound. But interestingly, when you have independent tests, then probability of type 1 or of Bonferroni is actually very close to alpha level when p is large. So this is like a simple calculation which show this when your alpha is 0.05, when you do 30,000 tests, like what we do often in, sorry, it should be p, not n. So p is 30,000, so which we often have in the genetic studies. If you do Bonferroni, if tests are independent, then your probability of type 1 error is very close to 0.05. Now the question is it a good test? So, there are many scenarios which can be behind your data. One of them is the needle in haystack. So we can imagine that all hypotheses are true apart from one. So only one mu is different from 0. And now, if you calculate this quantile of normal distribution, you can find out, so here is the critical value, this quantile, you can find out that asymptotically, it behaves like the square root of 2 log p. So indeed, it is of a magnitude so as to separate the noise. And it's very easy to show that if Bonferroni test is going to identify that there is a signal in your data, if a needle is slightly larger than this 2 log p, and it is not going to identify the signal at all, if your needle is of a length which is slightly shorter than the square root of 2 log p. And now the question is, can we do better? Is there a procedure which can find a shorter needle? I'm sorry, yes, sure.
Yeah, I have a question. So actually, I don't get the initial problem statement because as I see, well, we can run the test once and then just run it on different data sets. So see if it, if it matters or not. I mean, so, yes, yeah, you test significance. Why don't we test one more or two more times and check for statistical significance again?
You mean for the you want to repeat the whole procedure again and again?
No, just on a different data set. So we catch the significance, and if it was like a random thing, we can just.
Well, clearly you can do this. Okay, if you can do this, then you can do this, but often you cannot. Okay, so like in some cases the measurements. So indeed, very often what we do. So let me just put it this way. Indeed, if you look at a specific data set, you can spend a lot of effort. Very often, what we give a practitioner after such an analysis is some set of hypotheses which we rejected, and they are going to verify this. And this is not necessarily going to be a statistical test. They are really going to do biological experiments to find out if what we found out is good or bad, okay? But there are also situations like in astronomical studies. So there are many situations in which you can use such tests. Like in astronomy, very often you don't have data to verify the hypothesis. And in astronomy, they actually quite often test for such global null hypothesis. So depending on the data set, sometimes you have a chance to repeat the measurement. Sometimes the repetition, if you want to use a better technique, it costs a lot of money. So we really aim for trying to get to perform as good as we can doing just one test, okay? So what I want to tell you is basically, so the important thing is that when you have a large database and you look for patterns, you are going to see many random patterns due to the random fluctuations. And you have to adjust your statistical procedures for this. If you don't want to catch noise, like in the Bonferroni correction, then your critical value needs to be like the square root of 2 log p. If you don't have this like this, you are going to see many false discoveries. So verifying this would be basically out of, okay. So we are going to approach multiple testing in a moment. You will see that there is a problem, okay? Okay, so basically this is what you need to do. So if you want to separate the noise from the signal, you need to use the critical value at the level square root of 2 log p, and then you are able to catch only needles which are as long. Okay, and I'm going to tell you a little bit about how the statistical theory develops. So what people do to find out if there is something better. Is it really a limit of a multiple testing or of this global procedure? So what people do, I don't know if you know Neymann's tests. So if you compare two distributions, one is called H0 and one is called LA. Okay, so we have L0 and LA. L0 is your null hypothesis and LA is your alternative hypothesis. So if you know exactly these distributions, we know how to construct the optimal test. So the optimal test is basically based on the ratio of these densities, LA of X over L0 of X. So this is the optimal test. You cannot, so this is the Neyman-Pearson test. There is an easy proof to show that this is the optimal test. So you find the statistic, then you find the critical value. So the probability that it is exceeded under the null hypothesis is 0.05, and you cannot find anything better than this. So we have a Neyman test. We need to construct the Neyman test for the needle in haystack problem. So for this purpose, you need to basically find the alternative which would model the needle in haystack. And the alternative is such that we assume that there is a needle in my mu. So under alternative, there is a needle. The needle is of a length mu. So the test knows the length of a needle, but doesn't know where the needle is. So the distribution under the alternative is like a mixture of these p distributions which place needle at one of these positions. And now when you have this alternative, you can actually calculate the Neyman-Pearson test. This is the statistic, so I'm not going to go through the derivation. What I wanted to tell you is that this optimal test, the best test which you can create, is still not capable of identifying needle if this is shorter than the square root of 2 log p, asymptotically. So the Bonferroni correction, in a way, provides the optimal detection threshold in the situation of when you have a needle in haystack. So, but you do not necessarily need to have a needle in haystack, okay? So sometimes you might have many X's which are not necessarily very large, but which are actually, many mu's are different from 0. And another test which you can use is the chi-square test. And the chi-square test is simply sum of your xi squared statistics. And the global null hypothesis is true. This is going to have a chi-square distribution with p degrees of freedom. So it's very easy to pick the critical value. We know how we know that this is going to control the type 1 error at the level alpha. And then again, sorry, I'm going to skip all these. So here are very detailed calculations, which are kind of show that asymptotically for large p, the distribution of our, so T statistic, we normalize the chi-square distribution. We normalize chi-square distribution by subtracting expected value divided over standard deviation. And by the central limit theorem, under the null hypothesis, this is going to converge to a standard normal distribution. Now, under alternative, it's going to converge to the normal distribution where the expected value is equal to mu square to the square of L2 norm of mu divided over the square root of 2p. So we can see that this test will be able to identify the alternative if the L2 norm of mu is larger than the square root of p. Okay, so we have a different detection threshold, and again we can show using the again. Sorry, I'm going to skip this. I'm going to give you the slides, and please, if you are interested, there is a very nice presentation in the manual content that you have all of this proved. So you can also construct the Neyman-Pearson test for this problem. Neyman-Pearson test would know the norm, L2 norm, but wouldn't know where the signal really is in direction. So in the Neyman-Pearson test, we are using the distribution which is uniformly distributed on the sphere. But we assume that mu is uniformly distributed on the sphere of radius rho, which is and basically, we can show again that this best test is not capable to identifying the signal if the square of the L2 norm is smaller than the square root of 2p. So now we have two different tests. One is Bonferroni, one is chi-square. And the situation is that none of them is better than the other one. So the Bonferroni test will be able to identify just one needle. If it is larger, let's say, if this needle is of the length 2 square root of 2 log p, Bonferroni will be able to find it. The chi-square test will not be able to find it asymptotically. And on the other hand, if I have needles which do not increase, so they are all equal to 1, but their number increases with p, then the Bonferroni will be very weak, but the chi-square test will be strong. Okay. And again, now I'm going to skip a part of the lecture because I don't want, so I think I'm going to share the materials with you so you will be able to, this is here the discussion of the situation, what happens in the middle. So the general conclusion from this discussion is that the detection threshold depends on the density. Beta is a parameter of a density when beta is, so actually the parameter of a sparsity. So when the beta is close to 1, it means that the probability that the fraction of alternatives among your test is like 1 over p. So on average, you have just one needle. When beta is small, it means that you have more needles. Like when beta is 1/2, it means that you have like square root of p needles. So when you have more and more needles, they can be shorter to be identified. Okay, so that is maybe to expect it. Okay, but then you have to use specific tools. So if you want to be somewhere in the to perform somewhere between the needle in haystack and the many small needles, there are some specific tools like higher criticism test, which I'm not going to discuss. So just the lesson which we should learn from this. So the length of your signal needs to increase with p if you want to separate the signals from noise. So the more features you have to use the more stringent threshold to separate the signal from noise. The length of sparse needles to be detected depends on the number. If there are more of them, they can be shorter. And there is a single optimal method for testing the global null. The selection of a method should depend on the expectation on the signal sparsity. So in the high dimensional statistics, this is no longer true that we have, let's say, uniformly most powerful methods. So which method is the best, a little bit depends on how your data look like. Okay, and now I'm going to proceed to another topic, which is multiple testing. And in multiple testing, we test now not the global null, but we test each hypothesis separately. So we do separate tests for each mu I. So I have many genes. So this is like a real practical situation which I meet. So the practitioner biologist comes to me, shows me the table of expression levels, and wants me to point out at hypotheses, at genes which might be important. Okay. So this is a table into which we write down this analysis. So we do p-test. P0 hypothesis might be true. And we reject some of them. So R is the number of rejections. W is the number of acceptance. V is the number of false rejections. We call it false discovery. So the hypothesis is true, the gene is not important, but I'm saying it's important. So this is a false rejection. And now T is the number of wrong decisions of the other type. So the gene is important, but I accepted hypothesis. I haven't found out that the gene is important. So when you do such a test, so you pick any test you want, okay? T test, whatever, you pick some critical value. Now all you know is the values which you have in the last row. So you know how many tests you made, you know how many rejections you made, you know how many accepted you made, but you have no idea how many hypotheses were true. You have no idea how many mistakes you make. So we are trying to construct the multiple testing procedure in such a way that we control number of false discoveries in some way. So type 1 error is a probability that I reject the null hypothesis when it is true. So this is, I don't want to make false claims. I don't want to reject some well-grounded belief based on a noisy data. So I want to limit the number of false discoveries. And the measure of a type 1 error, like the classical measure of a type 1 error in multiple testing, is called the family-wise error rate. So family-wise error rate is a probability that I reject, that I made at least one false discovery. This is a criterion which was used for many years in multiple testing. So multiple testing started to develop in the 60s. And for 30 years, people were concentrating on this specific measure. But now when we do million tests at once, like when we do genetic studies, this turns out to be a very stringent procedure. If you want to control family-wise error rate, usually you have to pick a very conservative critical value and you have low power. So in 1995, people introduced another concept, which is called false discovery rate. So when you control false discovery rate, you are saying, okay, I don't care, I can make some false discoveries, but they need to consist a small proportion of all discoveries. So I want to control the expected value of a ratio. This is the number of false discoveries over the number of total discoveries. So this is FDR. Okay, so and now coming to your question, okay, so let's say that I'm making 20,000 tests at once. So I'm making actually 5,000 tests at once at the significance level 0.05. All hypotheses are true. And then I'm making 250 false discoveries. And believe me, if I go to biologists with 250 discoveries, he has no money to check all of this, okay? So this is like, we really need to try to reduce the number of discoveries. And again, the classical procedure to control for the number of false discoveries is the Bonferroni correction. And the Bonferroni correction, basically, you perform each test at the level alpha over p, okay? And again, it is, okay, so this is what you do. So you use basically this threshold square root of 2 log p for each of them. I don't know. So, okay, so maybe I will just continue for 3 more minutes and then we make a break. And now, in 1995, there appeared a procedure which turned out to be like a breakthrough procedure, which is called Benjamini-Hogbert procedure. This procedure is a bit more complicated. So what you do when you use this procedure, you sort your absolute x's from the largest to the smallest. And now you compare each of them with the quantize of a normal distribution. This alpha I value increases with I proportionally. It's alpha I over 2p. So let me just show you on the plot. This is these are the critical values which I'm using. So for the largest one, I'm using this critical value. For the second largest, I use a value which is smaller, and so on. And now I catch the last point here, according to the sequence, which is above the line. And I reject all hypotheses to the left-hand side. So this is called the Benjamini-Hogbert procedure. And here, yes, question?
I really Can you tell what is X and what is X?
So we again test the null hypothesis that so we are still using our model. So our model is such that xi, sorry, I don't want to come back. So xi comes from a normal distribution with mean mu I and variance 1. Okay, so xi is a statistic which you can obtain by a t-test or by what. So there are many statistics which you can transform to this form that it will have asymptotically normal distribution. So xi is a statistic which so this is a statistic which you obtained after transformation. So you can assume that it has a normal distribution with mean which is mu I and variance equal to 1. And you want to test the hypothesis that mu I is 0. Okay, so for each of these tests, we test the same. We test the hypothesis that mu I is 0. Okay, so each of these tests is the same. So we are testing, is this gene important or not? Or the second test for the second gene, test number 3 for the third gene. Okay, so we test like one after another one. And now this is interesting. Okay, so you do not use the same threshold for every test. You decrease the threshold. Okay, so this is a very funny procedure. It might look like. Okay, so this is not like in Bonferroni that you have some critical value. You sort your x's from in absolute value from the largest to the smallest. So our goal is to control false discovery rate. And this procedure controls false discovery rate. So why it works this way? So maybe let's give you a little bit of intuition. So we want to control false discovery rate. So we are basically saying that I'm going to accept false discoveries as long as they consist a small proportion of all discoveries. So when I discovered some genes which are really important, like these ones here, which are important, I can decrease the threshold a bit and let some noise into my data. So I am on purpose entering a little bit of noise because I can do it, because I want to control false discovery. So I can have a little bit of noise. I can have some V which is larger than 0, as long as V over R is small. So if my R is large, I have many important genes, I can let a little bit of noise. Why do we do this? Why do we let the noise in? Well, because when we do many tests, where the data are noisy, we have a low power in general. So by changing these critical values, when they become smaller, you can actually identify more genes. And what is interesting is that it is a nice procedure in a way that it controls for discovery rate. So there is a proof, mathematical proof. And again, if you go through the lectures of Emmanuel Kant, this proof is really not difficult. I think it should be at the level of the students. It's like one-page proof. We can show that if our tests are independent, so let's say genes are independent, then this procedure controls for discovery rate. So it is justified. So maybe I will just stop at this point. So let me just comment only on this formulation. So this procedure controls for discovery rate at the level alpha, P0 over P. We don't know P0, okay? We don't know how many true hypotheses we had, but we know that P0 is smaller than P. So we know that FDI is controlled at the level which is smaller than alpha. Okay, and I guess according to what I learned from the organizer, I am supposed to make a short break right now. Am I correct?
Yes, we can take short break, but if you want, we can not doing some break now.
Okay, so this lecture is supposed to be like 70 minutes.
Okay.
Yeah, this is. I'm just asking how much time I can take.
We can take 5, 10, 15 minutes break. So
okay, so maybe we do it as it was planned. So maybe we can make a 10 minutes break, okay. and we meet 2:15.
Okay.
Okay.
Yeah.
Thank you.
Thank you too.
Thank you.
Yes, yes.
Okay. So, so you had 10 minutes to rest and maybe hopefully think. If you have any questions now, I will be very glad to answer. Okay, so if there are,
yeah.
Yes. Yeah.
I think maybe later someone can ask something.
Sure, no problem, no problem. Yeah.
Okay. Thank you.
Okay, so we have these two procedures. So I told you that this Benjamini-Hogbert procedure for multiple testing, it can control for discovery rate, and it is like an imaginable success. So because people are now really doing millions of tests at once, and Bonferroni correction is very conservative. So if you use Bonferroni correction, if you don't want to have any false discoveries, you have to use a very stringent threshold. This procedure of Benjamini-Hogbert become very popular. So it is probably one of the most highly cited papers in statistics in recent years. So this theorem, which you can see, is under a pretty stringent assumption that X's are independent. But this can be actually weakened. So, sorry, I have some problem. But there's a new theorem, not new, but Benjamini and Yekutieli proved that when the test statistics are positively correlated, then you still control FDR. And if you have no idea about the structure of correlation between your X's, you can adjust Benjamini-Hogbert in such a way that instead of alpha, here you have to have instead of dividing by 2p, you have to divide by 2p times the sum, which is like log p. And then you control FDR independently on the structure of correlations. Okay. Okay, so now the question is, okay, we want to control FDR, let's put it this way, and many people want. And this is what people often do now in biological studies when they do many tests at once. But the question might be, is there any other reason for which we would like to control for discovery rate? And let me just think now, change the topic a little bit. Now we are not interested in testing. All we want to do is to estimate expected values. We want to estimate the vector mu, okay? So let's say that, so I wrote sigma square, okay, let it be sigma square. So we want to estimate vector mu for each, so xi, please remember, xi is already a statistic, so we already used some observations to calculate this. And now, in this specific problem, for each I have just one value xi. And in this situation, the maximum likelihood estimator, or the best estimator of mu I, is simply xi, okay, because the distribution of xi oscillates around mu I. So this is the maximum likelihood estimators. So what is the mean square error? So what is the mean square error? This is the expected value of the L2 norm of the distance between estimator and the vector mu, the true value mu. Well, this is some, here you have, here is the sum of expected values. You replace mu I with xi with a sigma square, you get p sigma square. So this is the average mistake you are making, when the mistake is the squared of the L2 norm between mu hat and mu. Can we do better? Okay, so for many years, people believe they cannot do better. So like it is well known that if you want to estimate expected value in the normal distribution, you cannot do better than by calculating the average or the. But it turns out that in large dimensionals, when p is larger than 2, then actually you can do better. So first thing is, I don't know if you know this formula. So what is the mean square error on the coordinate I? So you want to estimate mu I. Mean square error is the expected square of is the expected value of a squared difference between mu hat and mu. And this is the same as the square of a bias plus variance of mu I. So bias is the difference in the expected values between your estimator and mu. And variance is simply a variance of your estimator. So mean square error depends on the bias and on the variance. And in this situation, maximum likelihood estimator is the best among unbiased estimators. So if you don't want to have a bias, if you want the expected value of your estimator to be equal to μi, then you have to use MLE, maximum likelihood estimator here in this example. But it turns out that you can actually, so in our problem, as I say, expected value of estimator is equal to mu. Mean square error is simply equal to the sum of variances, and we get p sigma square. Now, a question is, can we improve our mean square error by introducing some bias? So what do we do? Well, let's consider a very simple estimate. Let's multiply our maximum likelihood estimator by some constant, C. Okay, so now you can calculate the bias and the variance. You can calculate mean square error. It is a function of just one thing, C. And now we want to find C, which minimizes mean square error. This is a very nice quadratic function, so it is enough to calculate derivative compared to 0. And you can find out that the optimal C is smaller than 1. So there is an estimator which is better than MLE, and the constant is equal, depends on the norm of mu. So the constant which you should use here is, as you can see, in the numerator I have the squared norm of mu, in the denominator I have the squared norm of mu plus p sigma square. You can do also another thing, and here is the difference. So here is the mean square error of your maximum likelihood estimator when p is 100 and sigma is 1. And now if you use this improved estimator, your mean square error can be very close to 0 when your mu is 0 and is systematically below the mean square error of maximum likelihood estimator. So you introduce a bias, you have a bias, but your variance is c squared times smaller than for the maximum likelihood estimator. And in the result, you get something which is better in terms of a mean square error.
Professor, excuse me, I think I didn't get the graph. So you say we set C as 0, right, in this graph? Oh, is it? Oh, yeah, that's fine.
C optimal. It's C optimal, okay?
Yeah.
Another version of this estimator is such that you can, but maybe I will just, so another version of this estimator is that you do not, so what does this estimator do? So this C is, this constant is smaller than 1. So this estimator shrinks, we call it shrinks, maximum likelihood estimator towards 0. So multiplied by the constant which is smaller than 1, which means we shrink towards 0. Here is a version of this estimator which does not shrink towards 0, but shrinks towards the average of all x's.
Professor, but this is the thing, exactly the same thing which we do with this corrected sample variance, right?
Sorry, I don't understand. What are you asking?
I mean this procedure we do it with the corrected sample variance.
What do you mean corrected sample variance?
I mean...
No, this is not the correction of a sample variance. No, we don't have sample variance here at all. Okay.
I mean, there are two estimators for one of them.
No,
and second has a smaller MSE.
This is true, but yes, this is true. So in a way, it is similar.
Yeah.
In a way, it is similar, but because this procedure, when you calculate the sample variance, again, you have two procedures. You have an unbiased estimator, and you have, let's say, MLE estimator. And I think the unbiased, in some circumstances, can have a larger mean square error than the biased. So this is similar in a way. The problem is completely different now, because I'm not estimating variance. I'm estimating mu.
Yeah, of course. I mean, the procedure, like the idea of what we are doing and why
yes,
we're doing that.
Yeah, this is true. This is true. So the idea is the same. So we are introducing some bias and reduce a variance. So we are going to have a smaller MSE. And let me just tell you that, okay, so let me just go on and I will comment when I show you the final result, okay? So here's another version of what we can do. You do not have to shrink towards 0. You can shrink towards the average of all your x's. And again, you can find the shrinkage constant in here, and this is the formula for the optimal D. The formula for the optimal D depends on the ratio. It depends on the variance, on the differences between the mu values in your sample. And again, this D optimal is larger than 0, which means again that this optimal selection of D is always better than D equal to 0. It's always better than in case when you use MLE. And again, you have this graph which shows that when you pick D optimal, you can get much better variance. Okay. Now, what is the hatch in here? Well, you cannot pick this optimal constant. You cannot pick this optimal constant because the optimal constant depends on the norm of mu, and you don't know mu. You estimate mu. But when you rewrite it in this way, you find out that what you have in the denominator is expected value of a norm of a square of a norm of your x. So you can actually estimate it, replace the expected value by the norm of x. Here is also a small difference, so we replace p by p minus 2, and this is what is called James-Stein estimator. So Stein was a famous statistician at Stanford. James was his PhD student, and then came up with this estimator. So and they have shown that when you multiply x by this constant, you are systematically better than in case when you use MLE. Okay, and this is an estimator. You do not have to know your mu. You can calculate this constant and apply it. So this is called James-Stein estimator, and it was a discovery. So it was like, I think it was like, as you can see in 60. Up to this point, people believed that when you estimate mu in the multivariate normal, it's the best to calculate simply the average, like in a one-dimensional case. And what James-Stein has, what they showed is basically that these two estimators are systematically better than MLE. So for whatever mu you have, they are better, which means that the maximum likelihood estimator is inadmissible. It just shouldn't be used. Okay, and like a short comment, the proof of this is very simple. You can find it in the lectures of Emmanuel Candes, and it's based on the Stein's identity. So Stein's identity gives you the formula for the expected value of the square of this difference in case when this estimator is equal to x is MLE, plus some g of x function. So g of x has to be differentiable, as you can see. So I need to differentiate it over all coordinates. There are some additional but very mild assumptions on g of x. And under these assumptions, you can calculate the risk using this formula, which you can see below. And this is not difficult. And if you use this formula, you can actually prove this James-Stein inequality. Okay, so what I have shown you is that in large dimension, we see some phenomena which we don't see in low dimension. So like when you estimate the vector mu in here, it's good to introduce some bias, reduce variance, and then you get a better mean square error. It turns out that in case when you are dealing with a situation when mu is sparse, what does it mean that mu is sparse? It means that mu has many zeros. When mu has many zeros, you can do even better when you combine estimation of mu with a multiple testing procedure. So let's say that you take your x values, you test which mu's are 0, which are not. If you accept the null hypothesis, your estimator is going to be 0. And only in case when you reject the hypothesis, you estimate your mu by x. And now there is a very famous result of very famous authors, like they are all very good, very famous, Abramovich, Beniamini, Donoghue, and Johnston. So Donoghue and Johnston are from Stanford University. Abramovich and Beniamini, they work in Tel Aviv in Israel. So they have shown that if you use Beniamini-Hogbert procedure as the test here, you can actually get asymptotically optimal prediction of mu. In case, so the assumptions which we are making is the assumption of sparsity. So we assume that when p goes to infinity, then the proportion of alternative hypotheses among all converges to 0, but it can converge very slowly. Okay, so there is a reason for which we are interested in the Benjamin-Hogbert procedure, not only because it controls false discovery rate, but also when you want to estimate the vector of mu, it can give you something which is roughly optimal. So just to tell you that there is some work which also I did in here. So sometimes you can think about testing as about a classification problem. So like regular classical testing is very asymmetric. So you assume that you have a null hypothesis, and it's very difficult to reject the null hypothesis, okay? So you have to, probability of rejection when the hypothesis is true is only 0.05. So the classical testing does not treat the null hypothesis and alternative symmetrically. But you can also think about testing in terms of classification. So you have two things to pick from. So you have alternative and null hypothesis. And you might have some loss. You might have some loss. You pay something if you make type 1 error, and you pay something if you make a type 2 error. And now let's assume that let's assume that my mu's come. This is a mixture. So what does it mean? It means that among my mu's, epsilon are different from 0. This is a percentage. And 1 minus epsilon are equal to 0. So 1 minus epsilon percentage of my mu's is equal to 0. And epsilon, we call it a sparsity parameter. And epsilon mu's are different from 0. And when they are different from 0, they come from the distribution which is normal with variance tau square. Okay, so a little bit maybe complicated, this model. But now, if you, under these assumptions, you can calculate the Bayes classifier. So you can calculate the procedure which minimizes expected value of a cost, expected value of a risk. And this is simply the ratio. Again, this is like a Neyman-Pearson test. So under alternative, when my mu comes from such a distribution, n0 tau square, when my xi comes again from the normal, but with variance equal to sigma square plus tau square. So this is the density of my x under the alternative. What you have in the bottom is the density of my x under the null. Under the null, mu is simply equal to 0. My x has a normal distribution with mean 0 variance sigma square. So again, like in the Neyman-Pearson test, I calculate the ratio of the densities. But now the critical value is selected not as to control probability of type 1 error, but is selected as to minimize the cost of the experiment. So it depends on the ratio of the costs for type 1 error and type 2 error, and also it depends on the proportion of the alternatives and null hypothesis in our sample. And we know, it is very actually easy to prove that if you use such a critical value, you are going to minimize the cost of your experiment. So what is the problem? The problem is that I cannot use this formula because usually I don't know tau, I don't know epsilon. Okay, I don't know tau, I don't know epsilon. So people are trying to construct procedures which could be used on the data, use the information in the data, and such that would approximate this Bayes classifier in terms of a risk. So we want to have the same approximately cost as for this optimal procedure, which is depending on things which we do not know. And these are the results which I did, and I did it with several people. So at first we did empirical studies. So these two papers which you are seeing here, we did empirical studies where we actually compared Benjamin-Hogbert procedure to many other procedures in terms of a Bayes risk, so expected value of a cost. And then we actually proved. So we proved that under certain conditions, Benjamin-Hogbert procedure allows you to achieve this optimal cost. So this is a paper which appeared in 2011 with the last person is Jayanta Kumar Ghosh. He is a famous statistician. He worked at Indian Statistical Institute, but also at Purdue University in United States, where I have known when I got to know him. Arijit Chakrabarty was his PhD student. He's currently working as a professor at India Statistical Institute, and Florian Fromled is my colleague from University in Vienna. So just to tell you that, these two results show that Benjamini and Hogbert procedure is interesting for many reasons, not only to control for discovery rate, but also if you want to minimize the estimation error of your mu. And if you want to optimize your base risk, it is good to use this procedure. Okay, so this is, I think, here are facts to remember. So in high dimensional problems, unbiased estimators can often be improved by biased estimators with reduced variance. When P is larger than 2, then the maximum likelihood estimator of a vector of means with independence is not admissible. It can be improved by James-Stein estimators. In case when the signal is sparse, this can be further improved by thresholding rules. Thresholding rules basically means that I decide by some test that I'm not going to estimate some mu because they are going to be equal to 0. And again, if you use a hard thresholding estimator of mu using Benjamini-Hogbert multiple testing procedure, this procedure adapts to unknown sparsity. So this procedure adapts to this unknown epsilon and is asymptotically optimal in the sense discussed in Abramovich et al. Or in our paper from Analysis of Statistics. Okay, so I guess this is all what I wanted to tell you about the multiple testing procedure. And now I would proceed to the supervised learning, so to the multiple regression actually.
Professor, just a small question. So here's this first point about that, well, introducing bias, we can reduce mean square error. And can you give some kind of intuition on that? Like why and, I mean, yeah, why and how? Like what's happening in that?
Well, you see it here, okay? So here. So this derivation tells you more or less everything. Okay, so when you use your variance, if you use the maximum likelihood estimator, your variance is equal to sigma square. Okay, because your xi is having the normal distribution. So your xi is having a normal distribution with mean mu I in variance sigma squared, so this is equal to sigma squared. So variance is equal to sigma squared. Now, when I multiply MLE by C, my variance is going to be C squared, sigma squared. So if your C is smaller than 1, you are reducing variance.
Yeah. Well, I see the derivation and understand how. I mean, I understand why mathematically, but maybe in terms of pictures or anything.
Okay, no, sorry, I don't have a picture. This is, some people say that this is due to the properties of the L2 norm. So
maybe it's because some distributions in there are not symmetrical, so you really benefit from like.
This distribution is completely symmetric. Okay, it's symmetric ground zero. Oh, and maybe this is a reason, a kind of. So you, sorry, let me just think for a moment. So the distribution of your xi is actually concentrated around mu I. Okay, so it seems that would be sorry, I didn't think about it for some time. So geometrically, I think this results from some properties of the L2 norm. Okay, so the L2 norm penalizes a lot things which are far away from you. So variance, which means that in some cases, in some instances, you are going to, when you change, how does it look in terms of plots? Okay, I could actually show you a plot, but you would have to wait for a moment. So I have to.
If it's not a problem, I just.
No, so just a moment. Okay, so I'll just show you more or less how it might look like.
Yeah. think. So again, it's symmetrical, and we're not benefiting from the.
Okay, so just a moment. I'm just.
Okay.
I'm back. So sorry, I
No,
was
I think I
away.
get it. So it's symmetrical, and we do not benefit because of the distribution is like kind of weird, and so we don't benefit from the fact that we just shifted to the favor of like more observations, but we do benefit from the fact that it's really like jumps a lot, and we reduce that, right?
You see my whiteboard now?
Yes.
Whiteboard, so I can write. Okay, so let's imagine a situation such that you want to estimate your mu. So we really estimate your mu. Okay, so this is your mu I, and this is the distribution of MLE.
Oh no, Professor, we see the PDF viewer.
Okay, so let me just see what I can do. So I'll stop sharing, and I will share again.
Yeah, good now.
Okay, so yeah, you can see right now the green stuff.
Yeah.
Okay, so you want to estimate your mu, and your mu I is somewhere in here. And now your MLE has a large variance. Okay, so the average distance will be like this. And now you create something which is slightly more biased, but has a much smaller variance. Okay, so this is your true mu I. This is the mu I of, this is expected value of the estimator. It's slightly shifted, but now the average distance, well, will be actually shorter. Are you with me?
No, actually no, because as I do remember, we shifted a bit left, right? Because we multiply by something which
okay. So if you want to see the situation which we currently have, this is what we do. So you are correct. We shifted towards left. So we have a mu I which has a large variance. Now I slightly shift towards left, and I have a much smaller variance.
So it's a bit, yeah. So a bit smaller variance, but we benefit from it a lot, right?
We benefited from it a lot. So it has a much smaller variance. So basically, if you calculate the distance, well, the distance can be large in here, but it's still much smaller, like when you do your MLE and you have such a distance.
Professor, excuse me. I guess we're not seeing what you draw just now, but I get it. But I get it. So that's cool.
Okay.
Yeah. Thank you.
So I need to stop sharing, and I need to start sharing again. Okay. Okay, so I guess, how much time do we have? I still can teach, I think, for like 5 minutes. So let me just start the new topic. Do you see my second set of slides?
Yes.
Yes, yes.
Regularization techniques. Sorry, you see now with this first slide, which is always wrong here. Multiple regression.
Yeah.
Yeah.
Okay. So, so we are now talking about multiple regression. So we have a situation when you have multiple regression. So we want to, and let's consider at the beginning very simple situation. So our n is larger than p. Okay, so this is a classical situation. Why this is it? Sorry, I have some problem with my screen. Is it okay? Can you hear? Can you? Okay, good. You hear me?
Yeah, yeah.
So we calculate our estimates by the least squares. So you minimize the sum of squares of we minimize the sum of squares of a difference between your y and x beta square. And this is a formula. I don't know if you know this multiple regression formula. You know it?
It's ALS.
Yeah, so this comes from so this is a very easy to obtain if you think about it this way, that when you want to minimize the distance, you need to project your y on the space spanned by the columns of x. So you get this formula for the least squares estimators. And now, because in this classical regression model, we assume that x is fixed, x is not random. So this x beta term is simply like the expected value. And all the variability which you have in this model come from these error terms. So we assume that the error terms have a normal distribution with variance sigma squared via independent. So now, with again a quite simple calculation, it turns out that this least square estimator has exactly the multivariate normal distribution. Okay, you know what is a multivariate normal distribution, or should I say something? You know. So this is a vector mu, and here is a covariance matrix. This is a covariance matrix. This
Actually,
is
Professor, I bet that maybe some other students would say. I mean, because.
Okay, so please ask questions. So I have lots of materials which is very much advanced, and maybe it does not make sense if I proceed too quickly.
Everything is clear up to the point, so please proceed.
Okay, very good. So with the covariance matrix, it's a matrix of dimension p by p, okay? X prime X inverse. And we estimate variance of our, so as you said, Pablo, okay, we estimate variance here by the residual sum of squares, which is the sum of squares of differences between what we observe and what we fit, and divide over n minus p, where p is the number of columns in our X matrix. And this is the unbiased estimate of variance. And now we do a t-test. So if you just do linear regression in any program, it's just going to calculate your estimates, calculate this covariance matrix. So on the diagonal of this covariance matrix, we have the variances of each estimator. So we can calculate standard deviations just dividing by taking the square root of this. So sorry, there's a mistake in here. It should be square root, okay? F of beta is standard deviation in the square root of this. And now, what problem do we have? Well, if you let your p to be large, then when you calculate this X prime X inverse, then you will find out that you will have very large element on the diagonal. So the typically elements on the diagonal of X prime X become large as p increases. So you can find it if you play a little bit, and I can tell you a little bit about some theory. So let's assume that your X's elements of your X matrix are IID from normal distribution with mean 0 and standard deviation 1 over square root of n. Okay, so this is, I'm doing this as an example because this is like, so this is a relatively easy example. Your X's are independent. So each of your regressors will be independent from the other one. And we know exactly that when X's have such a normal distribution, if we multiply X prime by X, we will have a matrix, which has a matrix distribution, which is called the Wishart distribution. You can go into Wikipedia and find out what it is, okay? So it has a Wishart distribution, and the element on the diagonal of this matrix will have the expected value equal to 1. Now, when you invert this matrix, it will have an inverse Wishart distribution. And now the elements on the diagonal will have values which are equal to n divided over n minus p minus 1. So now you can see when you let p increase, when p becomes close to n, then this element on the diagonal will become very large. So variances of your estimators are going to be very large. And what price you are going to pay for this? Well, your mean square error increases. So here we have a situation when I have n equal to 500, and I have only 10 important regressors. And I'm calculating how big error I'm making in estimating these first important coefficients. So initially my error is very small, but when I include in my databases many X's which are completely irrelevant, I pay a price. So when I add to my databases new X's, my mean square error of estimating these first 10 increases. Okay, variance increases. And when p is equal to 480, variance is just huge. immense. Okay, so your mean square error is very large. And also, here is a test. So I'm testing for these 10 first ones, if they are important or not. So if you have a proper model where you only have 10 x's in your model, the power is 90%. So probability that you are going to find these x's which are important is 90%. But when you have large database and over 400 of your x's are not important, and you want to find these 10 important, you do a test, then your power of your test is basically at the significance level. I hope you understand this. So we simply, what happens is when you increase your p, so your p becomes close to your n, so your number of columns, your number of variables becomes close to the number of observations, then the variance increases a lot, and the power of your tests drop down. So this is a price. This is a price. So if you don't do anything, you will basically lose the power of identifying important regressors. Due to a large variance of your estimators, you are not going to see important ones. You are not going to see them among many, many false ones which have large variance. I hope this is understood.
Yeah, this is understandable.
Yeah, this is so I think is it time, Isabella, to make a next break?
Yeah, I think we now can a 10 minutes break. And
okay.
Yep.
So this is the introduction. This is the introduction to tell you that we have to develop other methods. So testing, when p is large to n, testing using the full regression model is not a good thing to identify important variables. We need to look into other techniques to identify important variables. And this is what we are going to talk about in the next lecture. So thank you. Like 10 minutes break right now.
May I ask you to share the slides with us now?
Right now?
Yeah.
Okay, how do I do it? Do I send to you, Isabella?
maybe after lecture.
There is no problem for me. I can send them to you, okay, right now.
Okay, maybe.
On the slides, and I'm definitely not going to go over all of this, but at least you can see. There are also some references if you would like to read more. Do you have any questions about what happened so far? Okay, sorry, let me just share again. Okay, you see my slides now, do you?
Yes.
Okay. Okay, so we are now going to talk about model selection techniques, and let me just start from a thing which is called for model selection. This is a set of techniques to identify important variables. And clearly, you cannot use a residual sum of squares for this purpose. So residual sum of squares, so the distance between what you observe in the training sample and what you predict. So basically, the prediction error in the training sample is not a good. It can be used to identify to basically select between models of different dimension. But it cannot be used to select variables, because whenever you add a new variable into your model, residual sum of squares cannot go up, and in most of the cases it will go down. So the only situation when the residual sum of squares does not go down is when you have algebraic orthogonality between your new variable and your y, or when you have a linear dependence of your x to those x's which are already in the model. Otherwise, even when the x variable which you want to include has completely no influence on your y, your residual sum of squares is going to go down. So residual sum of squares is not a good criterion for model selection. Or if you are familiar already, if you already did something, so there is such a thing like R-square. R-square measures the percentage of explained variability. And again, R-square is not a good criterion to select between models of different dimension. So what could be the purpose for your building your model? Well, you might want to have a good prediction error. So let me define in this setup what I mean a prediction error. So let's imagine that I have the same x, fixed x is fixed, which I had in my training sample, but I now have a different sample of the errors. So let's say I have the same x, but I repeat my experiment, and now I have different epsilons, different error terms. And what I want the model be able to do, I want the model which is trained on the training sample to be able to predict well a new observation, okay, new y star. So my prediction error is the expected value of a square difference between a new sample with new epsilons and the prediction which is based on the training sample. So let's imagine, so what is mu? Mu is the expected value of our y. So our y, expected value of y is the same in the training and this new sample. And it is equal to x beta. X beta is a vector, it has n elements, and this is a vector of expected value of my y. So what is my prediction error? Well, my prediction error is the difference between mu and our prediction. So our prediction of mu is also y hat, okay? So this is the estimator of expected value of y. Plus there is n sigma square, which will never disappear. So n sigma square simply comes from the fact that even if I exactly knew x beta, if I exactly knew the expected value, or if I exactly knew my beta, I would still not be able to predict my y completely because this is this error term, this epsilon. And expected value of a squared norm of this is n sigma square. So this is how the prediction error looks like. So what is the difference between the residual sum of square and this? So in the residual sum of squares, instead of mu, you have your y. So a residual sum of squares measures the distance between what you have in your training sample and the prediction. And what you really want to have a small is like the difference between your prediction and mu, xb. Okay, so let me just show you the result. So the result is such that the result is a bit more general from what we at this moment need. So let's imagine that your model predicts your y using a linear operator on y's in your training sample. So y is your training sample, you make a prediction by using some M matrix. Okay, so you have some linear operator on your y. And it turns out that the prediction error of such an estimator is equal to the expected value of a residual sum of squares plus 2 times sigma square trace of this M matrix. And I'm not going again to go through the, so you have a proof in here, which is very simple and goes by Stein's identity, which I have shown you last time. But you can actually, there are several techniques for the linear operators. You do not need to use Stein's identity. When you use Stein's identity, the proof is very simple. So here you have a proof. I'm not going to go through all the details. Okay, so you kind of have to believe me. Okay, so now what happens? So this is the formula which I'm deriving. So prediction error is n sigma square. So what is the prediction error? N sigma square plus, okay, so expected value of residual sum of squares plus 2 sigma square trace of M. And now when you do least squares estimation, your prediction is a projection. Okay, when you build, when you try to minimize the sum of squares of residuals, you simply need to project your y on the space spanned by the columns of x. And this is a formula for the projection matrix. And we know that the trace of a projection is equal to the rank of this matrix X. So if the columns in your mixed matrix are independent linearly, then the prediction error is equal to the expected value of residual sum of squares plus 2 times sigma square p. Okay? So you can forget about the expected value and you can simply estimate your prediction error by adding 2 times sigma square p to your residual sum of square. And this is an unbiased estimator of your prediction error. And this is what you might want to use to select your model. So you might want to try to use this criterion, minimizing prediction error, to pick the model so as to have the best optimal prediction. Okay. And now it turns out that this criterion, minimizing prediction error, coincides with a very famous criterion used in statistics, which is called Akaike information criterion. So in this specific situation, they agree. So Akaike criterion also would recommend selecting a model for which residual sum of squares plus 2 times sigma square p is minimal. Okay. Okay, so leave one out, cross-validation, I'm going to miss. So you probably know a little bit, so you know about cross-validation. Should I say a few words?
Pavlo, Alexey, Fedor.
So you know something about cross-validation? Have you heard?
Maybe say
Yes, of course.
Sorry? Should I say something? So this is, I'm not going to use it later in the talk. So just very briefly. So
Okay.
you can also use cross-validation to estimate prediction error. Cross-validation works this way that you basically take your sample, split it into some parts, treat one part. So let's say you split your sample into 10 parts. One part is going to be a test set, and 9 other parts will altogether make a training sample. So you train your model on 9/10 of your data and use it to predict 1/10 of your data, and you measure what is the accuracy of your prediction. So this is a very tedious procedure because you have to split and repeat all procedure many times. But in case if you do leave one out cross-validation, so leave one out works this way that you take one observation, remove it from the training sample, fit the model on all the remaining ones and try to predict this one. So y hat I is the prediction which you made using the whole sample without this specific observation. And for this one cross-validation, there is a closed form formula which does not require repeating regression many times. But okay, this is just to let you know, and I'm going to concentrate on model selection criteria. So now we have a Akaike criterion.
Malgorzata, we have a question. Sorry, I didn't understand what is P. Small p.
P, okay. P is the number of columns. P is the number of variables in your model. So number of columns in your X matrix. Okay, so P rank of X is equal to P. P is the, always in my slides, N is the number of observations. P is the number of columns. This is not a very good notation here because usually I use P to denote the total number of columns in my database. And here, P is the number of columns used to build my model. So P is the number of variables which I use to fit my model, and I calculate residual sum of squares based on the model with P variables. Are you with me?
Yeah.
So here, so, and I'm very sorry, I'm in the future slides, I'm going to use K in here. K is the number of, for a specific model. Number of variables I use to build my model and calculate residual sum of squares. So when I include more and more variables into my model, this is a penalty for the model dimension. We call it penalty for the model dimension. P is the dimension of my model, is the number of x's. So when I increase the dimension of a model, my residual sum of squares goes down, but I have a penalty which goes up. And this sum has a tendency to go down initially and then go up after you actually create your true model. When you included all variables which are needed to predict your y, this is going to stabilize. You can also see from this formula that when you add new variables into your model which are complete noise, your prediction error is going to suffer. So your prediction error, this penalty, when this thing becomes increasing, when you already included all important variables, prediction error is actually going to go down. Go up.
Thank you.
Okay, so a few more words about Akaike information criterion, just for you to know, because I'm now concentrating entirely on the regular regression, but you probably know some other model like logistic regression, when your Y is binary. There are Poisson regression models. In these models, you don't fit your model using a residual sum of squares, you use the likelihood function. And just to tell you what is Akaike criterion in terms when you have a likelihood function. So let's say that you have a vector of independent random variables from some model, and the number of parameters in your model is k. Okay, so theta belongs to ak. So I'm calculating the likelihood function for this model. And now what is and now I'm comparing models of different dimensions. Like in regression, I'm adding new axes, so my beta vector becomes larger. But there are many other situations when you use this, like when you, for example, want to estimate the density using orthogonal series extension, you might want to include more and more basis functions. So you want to compare models of different dimensions, and Akaike criterion tells you that you should pick the model for which logarithm of a likelihood function minus the number of parameters is maximal. And there is a very nice justification for this. You should, if you are interested and you write me an email, I can send you some references for nice proofs. So this does not come out of blue. There is a theoretical justification for this. And basically, this criterion is aimed at minimizing the, we call it Kullback-Gleiber distance between the density, true density of your X and the density provided by your model, which you fit to your data. Okay, so this criterion is aimed at, in our situation, it's aimed at minimizing the prediction error. And I'm going to go just to show you this slide. So in our regression model, our independent variables are our errors. So you can build the likelihood function, calculate the logarithm of this, and you can find out this is the formula for Akaike which you need to maximize. This is the same as minimizing residual sum of squares plus 2 times sigma squared k. So sorry, I'm now going to use k. K is the number of variables in my model. Okay, p will be the number of variables in the total base, and k is the number of variables I use to build my model. Okay, what happens if you don't know sigma? So here you need to know sigma. Well, you can estimate sigma by the unbiased estimator, one way. Another way, you can calculate log of a likelihood function, and you also want to maximize this with respect to sigma. And if you do it this way, then you are going to end up with a slightly different form of Akaike criterion when you penalize not residual sum of squared by log of residual sum of squares. Okay, so these are just to let you know. Okay, and here is the example which shows that if you, I'm just comparing 5 different models. Model with 10 variables, which are important. Then I added 40 noise variables. So I have model with 50 variables, 200, 400, 480. I did many simulations, and in all these simulations, all these box plots, this is the difference in Akaike between a given and a true model. All these box plots are above 0. So in all the simulations which I did, Akaike criterion had no problem to tell me that I should use only these 10 variables. When I add 50, my prediction error goes up. If I add other 150, it goes even more, and so on. So when I compare these 5 models, no problem. Akaike criterion can identify the true model. And there are many, many situations nowadays where Akaike criterion is very useful, but it is not useful for selection variables from a database. What do I mean for selection? So here I just compared 5 different models. But when I select variables from the large database, then I'm comparing much more models, and Akaike is no longer useful then. So first problem you have is a discrete optimization. So So I have a formula for Akaike. I have to calculate the residual sum of squares and then the penalty. And to do this, to find the model which minimizes this Akaike criterion, I would have to calculate this criterion for all possible regression models. And now assume that I have p variables in my database. Okay, I have p variables in my database. So how can I code my models? Well, I can take a vector which has a length p, and I can use a binary coding. So basically, in a given position, ith position, I can add 1 when my variable ith variable is in the model, 0 otherwise. So the number of possible models, model is, I would say, in this situation, I think about the model as a selection of variables which I use to predict my y. So the number of models is the number of p-dimensional binary sequences, which is 2 to the power p. So if your p is 50, 2 to the power 50 is already a huge number. And on most computers, you would not be able to check all models if your p is 50. And I'm not thinking about p being 50, I'm thinking about p being thousands or million. So really, I have a huge problem. So I cannot really look over all possible models. So I have no guarantee that I can find a model which has a best value of Akaike criterion. Okay, so this is an obstacle, which, however, people are dealing with. So people developed many heuristics, which go and try to find the optimal model. And with heuristics, under some assumptions, usually depending on the problem, some heuristics are better, some are not, some are worse. These heuristics with large probability can identify models which are closest to being optimal. So such heuristic is, for example, a forward selection. So forward selection works this way, that you basically at first look at all variables which you have in your database and pick the one which gives the smallest residual sum of squares when you build a simple regression model with this one variable only. Then you fix this variable and you look for the second one. Again, you look through the whole database and try to find variables which, after you add it to this first one, again has the smallest residual sum of squares. And then you try to add variable number 3. So in this way, at the first step, you have to fit p models. In the second step, you fit p minus 1, p minus 2, and altogether you fit about p square models, okay, p square over 2 approximately. And p square is already a number which we can handle, even when p is like million. Okay?
So the problem reduces to sorting, right?
Yes. Well, not exactly, because when you do your, so if you do simple regression, okay, you find your residual sum of squares. Then you find the minimum one. Let's say you sort variables according to this first step. This order is not going to be preserved. So like, let's say you have x1 and x2 variables which are very strongly correlated. And they are also, and let's say x1 is a very important predictor. X1 is very important, okay, the strongest one. So when you do linear models with x1 and x2, they are both going to have good properties. x2 is not a good predictor, but it's very strongly correlated to the good one. So when you do the residual sum of squares, both these models are going to have very small residual sum of squares. So if you make sorting, then your x1 would be first, x2 would be second. Okay, but now you take your x1 into your model, and now you look for variable which improves x1 the most. And x2 is not going to improve x1. Okay, so in the second level, the sorting is going to change. So it's really not sorting. You really have to, at each step, look at all variables again. Are you with me?
Yeah.
Yeah, it looks like a sorting algorithm, but you cannot run quicksort on that. Yeah,
no,
you really need to compare it. Yeah,
it's not sorting really, because as I say, the order changes at every step. So it is like you have to find really minimum, which is easier than sorting, kind of. You don't. So the sorting which you create after the first step is going to change at every other step. So if you have some correlation between variables, then sorting really changes. So not sorting, but order really changes of importance. So like in multiple regression model, an importance of a given variable depends on what you have in your model already. So as I say, in the situation when I had this x1 and x2, x2 would be a very good predictor on its own. But when you already have x1 in your model, x2 is not important, because x2 is very strongly correlated with x1. So, linear regression is a very interesting topic, and realizing what happens when you have correlated predictors is there's very nice mathematics behind it. So if you are interested, then it's good to read some good book about linear regression, which explains a little bit of mathematics behind all of this. Okay, so
I'm sorry,
yes,
in our chat, one question. In case of forward selection, usually we try to choose variable which has the most correlation with residual, right?
This is true.
Yeah.
This is true. So this is true. So basically, you exactly this is what you do. So at first, your residual is simply the vector y, because you have nothing in your model. So you look for a variable which is most strongly correlated with y. Then you have your x1 already in your model. You calculate the residuals, and then you look for variables which are most correlated with these residuals. Then you take x2, calculate residuals again, and again look for correlation with these new residuals. So this is a very good description. Yes. And if you do forward selection, let's say, so we usually stop it when the value of our criterion starts to increase. But in principle, checking all models would require not all models, but creating the whole path would require only p-square steps. And p-square is doable already for very, very large dimensional situations. With current computers, we can handle, like thousands of variables with forward selection. What is the disadvantage of forward selection? Basically, if you have a complicated model, let's say when you have 50 variables which are important, at the first step, you are picking only one. And sometimes it happens that you don't pick any of these important ones, but only some other variable which is strongly correlated with linear combination of these first 50. So this is some problem, because initially when you feed your model, the model is never correct, because your correct model contains 50 variables. And at this initial steps, you can easily make mistakes by including variables which are simply correlated to some linear combination of important ones. So a better formula, better method is a backward elimination, if you can do it. So backward elimination requires that p is smaller than n. So you can feed the whole regression model. Let's say your p is 50, so you can do it. N is 2000. And then you eliminate variables one after one. So you basically eliminate variables which have the smallest correlation with residuals. But again, backward elimination is better in a way that we kind of assume that the full model is correct, so that the whole variables which are important are in our model, plus maybe some which are not important, and the coefficients are than 0. So in principle, statistically, it is more accurate, but you cannot do it when your p is larger than n. And there are combinations of these two, which are called stepwise selection. So you at first do forward selection, then when your criterion start to increase, you then do backward elimination step. And because of what I have told you, that the ranking of your variables changes after each step. So you change your residuals, so you change your correlations, and it changes after every step. This makes sense to alternate between forward and backward elimination. So if you alternate, so like in the backward step, you often remove variables which were included at the first steps of forward selection. So this makes sense. Stepwise selection is a better procedure than these both. So here I would like to recommend a library which was written by my PhD student in R language, which is called Big Step. So I do a lot of this type of research because I do applications for genetic data. And in the genetic data, we had to deal with like 2 million variables. So my former PhD student created an R library which can really handle many, many variables. And you can use this variable. And now we can try to use this AIC criterion, Akaiike criterion, to pick the best model when my database contains of 500 variables. True number of important is 10. So I simulated my data. There are only 10 important variables. And now I run my Big Step library with Akaiike criterion. And what I find out is that it selects more than 100 variables. So it does no longer work. Akaiike criterion goes into models which are much too large. Okay?
Yeah, Malgorzata, we have one question. In backward eliminator, we kick off variables by p-value.
Yes. So you see, like this is the same. Okay, so how are the p-values? So yes, so you can do t-test. You pick the one which has the largest p-value, and this is also the same, which will have the smallest correlation with residuals. Okay, so these two things are completely exchangeable. So so p-value is large. There is a small correlation between your y and residuals. And also, if you remove your variable, then residual sum of squared drops by the your residual sum of squares increases by the smaller amount. So you don't lose much in prediction when you drop this variable. Okay, so these are like three things which are equivalent. Yes, indeed, you can look at p-values and eliminate the ones which have the largest p-value.
Thank you.
Any other question?
In chat, no. Maybe someone want to ask something?
Okay, so what I wanted to tell you right now, what I'm showing you, so just a moment ago, I have shown an example when Akaiike criterion works very well. And now you can see that here, when we use it to identify variables in large database, it's just very bad. It includes 100 variables instead of 10. So I repeated here the experiment 1,000 times, and you can see that Akaiike never picked less than 70 variables. And now let me try to explain it, and you will find the relation between this and multiple testing, okay? And I'm going to explain it on a very artificial example, which is the simplest. This is the easiest situation for Akaiike and for any criterion. And this easiest situation is such that the X matrix is orthogonal. So X prime X is identity. Okay, so this is an artificial example. It is the easiest example. So now when I calculate my estimator beta hat, it is simply Y transpose X. So this is like the product of YX. Now let's calculate residual sum of squares. So here you have a formula for the residual sum of squares. And you can find out here in this situation when X prime X is identity, then the residual sum of squares is simply equal to Y prime Y minus sum of squares of your beta I square. Okay, your estimators. So basically, at each step, your residual sum of squares goes down by exactly by the quantity which is exactly bi square. So if you use Akaiike, so you are going to add a new variable if your beta I square is larger than this penalty for dimension, which is 2k. Okay, so beta I square has to be larger than 2 sigma square. Okay, are you with me? I hope you are with me still.
Yeah,
So
yeah.
basically, yeah. So if you want to include beta I, it has to be larger than beta I square has to be larger than the 2 sigma square. Beta I has to be larger than the square root of 2 times sigma. What else do we know? We know that when x prime x is identity and your beta I is 0, then your estimator has a normal distribution with mean 0 and variance sigma square. So you can actually calculate this probability exactly, and this probability is 0.16. So probability that that Akaike in the orthogonal situation, probability that Akaike is going to include a variable which is false is 16%. Okay? So when I have, question?
No.
When I have 500 variables, 490 of them are false, then I will have 78 false discoveries on average. And the typical size should be around 90 variables. Okay, so in my situation, the average number was 100 because, first of all, I didn't have orthogonal design. I had normal variables, so which were not orthogonal. And in everything, I used the criterion with log residual sum of squares with unknown sigma. But you see, these theoretical calculations actually show you what happens. So Akaike criterion has no multiple testing correction. So basically, the probability of including any false one is 0.16. So when your P is large, you are going to include many false discoveries. Okay. Okay, maybe this is going to be last slide. So there is another criterion, Bayesian information criterion, also very popular and also having nice theoretical justification. Here the penalty is sigma square k log n. So I didn't explain to you where it comes from, but it comes from Bayesian statistics. And again, it is a different threshold here. Okay, maybe this is a good point to stop. Okay, so this is like 35 minutes again. So like 10 minutes break. Okay, if it is fine with you.
Yes.
And
Okay.
we'll continue like 3:45. Okay.
Yeah. Thank you.
Thank you.
Thanks.
Yes, hello.
Okay, so let me just share the screen. Okay, so we already did this. Okay, so let me just go and recall my explanation when a Akaike criterion is not working. So I have shown you that the Akaike, if you have an orthogonal situation, then there is a fixed constant probability with which Akaike includes a given variable, which corresponds to beta I equal to 0, 16%. This means that this criterion is not consistent even in the classical situation. So when your sample size increases, it has no influence. So always you will have probability that xi is selected equal to 0.16. So Akaike criterion is not consistent in a way that probability that it will identify the true model is not going to 1 when n increases, when the sample size increases. However, in the situation when your p is fixed, asymptotically it will pick the model with best prediction properties. So the model with the best prediction properties might include some false predictors, okay? But in the situation when p is large, as you can see, there's a disaster. So here we have a second criterion, which is called BIC. And BIC has a completely different origin. It comes from the Bayesian statistics, and it is based on the approximation to the posterior probability of a given model. So in Bayesian statistics, we calculate such things as the probability that the model is true given the data. And BIC gives you the approximation to log of this probability. And the penalty here is a different structure. So instead of sigma, so you have additional factor, which is log n. And when n increases, this factor increases. And now when we repeat our calculations now, we will see that, for example, when it does not depend on p, again, this penalty, but depends on n. And when n is equal to 500, the probability that I'm going to include a wrong predictor is very small, like 1%. And also, as you can see, this critical value increases with n. So for any false predictor, probability that I'm going to include it in my model is going to converge to 0. So for any single one, which is false, shouldn't be in the model, probability that I'm going to include this is 0. So converges to 0. But we have many of them. So we need to multiply. So the number of false discoveries is equal to P0 times 0.13. And even in my situation when P is 500, even though this probability is very small for each single one, I'm still going to see on average 7 false discoveries. And when P0 increases, I'm still going to see more and more discoveries. So this criterion for each given variable asymptotically is going to give a true decision. But if you consider the number of false discoveries, is going to increase when P increases, because this criterion has no multiple testing correction. And here you have a simulation study which shows this. So again, we did a simulation, and you can see like that the average number of false discoveries really oscillates around 7. Okay, so BIC also does not prevent false discoveries when you look through a large database. Okay, so there is one more criterion which is much less known. Less people know this. This is called risk inflation criterion. And here the penalty depends on the number of variables. So you see our known factor 2 log p. So this 2 log p penalty is what we need to do multiple testing correction. And now when I calculate this, then probability that I'm going to select xi that beta, for which beta I is 0 is equal to this formula, more or less. And it's going to converge to 0 when p increases. So for this criterion, I have a multiple testing correction. And when p increases, probability that xi is selected converges to 0. However, the penalty here does not depend on the sample size. So for any given p, probability that your xi is selected is equal to something very small. And it's not going to small when n increases. So again, it lacks a little. So when n increases, when the sample size increases, typically we can get a very high power. And we could also have a small type 1 error. And this criterion does not do this. Okay, but this is much better. So the risk inflation criterion includes the correction for multiple testing. So it's much better in large databases. And now a few words about what I did. So this is a research which I did with two people. So you can see now Jayanta Ghosh. So Jayanta Ghosh was a professor at Purdue University when I was there. He unfortunately passed away 2 years ago. He is a very famous professor, so he had some very important contributions to statistics. And on the left-hand side is Professor Rebecca Dorch. When I met her, she was at Purdue University. She was working on statistical genetics. And she is currently a dean in the School of Science at Carnegie Mellon University in the United States. So we did this work together. So Rebecca is an expert in genetics and Jayanta Ghosh was an expert in Bayesian statistics. When I started to work with them, they asked, so Rebecca told me that she needs a criterion to identify genes, to localize genes. So the article appeared in genetics paper and is concerned with gene identification. So I don't have a slide about it, so let me just say a few words about it, how it works. So basically, when we want to localize genes, I have genetic markers. So like on the chromosome, there are positions at which I can genotype. I can check what is the genotype. And the genotypes can take only 2 or 3 values, which we call the 0, 1 or 0, 1 and 2. And then these are my X variables. And I have some Y variable, which can be like blood pressure. And I'm trying to identify these X's, these genes, which influence my Y. So this is like a regular multiple regression problem, and we needed to build a criterion. So you see, it was 2004, and the 2004 people still did not realize that BIC is not working here. People believed that in these types of problems, like in genetics, you can still use classical selection criteria like Akaike or BIC. And we found out that they do not work, that they include too many genes. So we come up with too many genes. And we came up with a criterion which is called MBIC, Modified Bayesian Information Criterion. So here you have a version of a criterion with log of residual sum of squares when the sigma is not known. And this criterion combines the penalty from the BIC and from a risk inflation criterion. Now here you have a constant which is called C, and now modified BIC is basically a Bayesian criterion. So it uses Bayesian statistics, and the C value has an interpretation of being the prior expected value of a number of genetic effects. So when you expect to have 20 important genes, you plug in 20. When you expect to have 10 important genes, you plug in 10. And you still have a penalty, which is going to increase with P. When you have more and more variables in your database, then you still need to include a penalty, which is like 2 log P. So the C constant should depend on your prior expectation. Now, let's assume that you have no prior expectation. So this is your first time you look at this problem. So we calibrated C. We calibrated C. So when this is not written here. So when you plug in C equal to 4, so we usually use this criterion with the constant C equal to 4. Then this criterion controls family-wise error rate with constant C equal to 4 for typical sizes used in genetics. So when N and P are both larger than 200, then this criterion in most cases controls family-wise error rate, so does not include false genes with large probability. Okay.
Professor, but it looks for me like here you really can benefit from that Akaike thing, because you know you from there you can get the estimate of the variables. You see, because you have.
No, Akaike completely does not work.
No, I mean, say you have 13% of including a variable into the model, right? So you certainly include the right ones, so 10, and then you include, I don't say 78 of the wrong ones. So say if you use Akaike and it tells you 88 on average, then you're like, okay, probably there are 10 of them which are the right ones. Isn't that the case?
No, 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 no. If I knew how many right ones I had, then it would be much easier to identify them. So if I knew that I only have 10 variables, I could basically look through all the models which include 10 and pick the best. I don't know how many of them are.
Doesn't it give you kind of an estimate for it?
We don't. So let me just okay. So just don't think about it this way. So I told you that she here has the interpretation of expected value by the way how it was derived, but we usually don't have any expectation. So usually you don't know what your C is equal to. Akaike is not really, Akaike is just completely wrong here. So Akaike just picks too many variables and this is it, okay? We want to have approximately the number of variables which is close to the true number. As I told you, if this criterion, to calibrate this criterion, we use the constant C, which is equal to 4. So when we plug in C equal to 4, then we have a multiple testing correction which controls family-wise error rate approximately at the level of 10%. Okay? So all these criteria, they are based on residual sum of squares, and you see no point to use, so it is much better to use BIC, because BIC gives you only 6 false discoveries, okay? And then it's just much easier to, okay, another thing which I told you, again, let's say Akaike gives you 80. So you have a very large variance for this model. Your P is 100. So even if I reduce the attention to the model selection by Akaike, I would have, so your P is 120. So if I calculated my estimates of beta HUD using this model, they might be not properly ranked because of a large variance. So BIC gives you a model with just 6 false predictors. So on average, there will be 20, which will have much less variance. So actually, based on this criterion, we could probably have a much better ranking.
Yeah, yeah. Well, generally, that was the idea, that we actually, from these kind of useless things, we can get something which is useful, which is the proxy for the amount of variables.
Well, you see, like, as I say, you are far away from being close to proxy when your P is large. So when your P is large, your Akaike is 10 times, it can be, you see, this is
yeah, I get it.
P0 times constant. So you are far, far away from the true number. And even BIC, here BIC, in this example, when P is 500, it gives only 5 false discoveries. But when P is like around 1 million, like what do we do? It is just horrendous. You have just you have models which are much too large. And I cannot go to genetics with a model where 90% of variables which I selected are actually false. I can come up with some false discoveries, but not with many. And here we have a linear dependency. So the number of false discoveries, sorry, let's look at BIC, is still linearly dependent on P. So when P is very large, I'm going to end up with a large number of false discoveries. So everything here depends on the ratio between N and P. So I'm now talking to you about statistics when my sample size is small compared to the number of parameters or compared to the size of a database. And I want to tell you that still you can do something. So P is much larger than N, and still we can do something, and we have to do something because this is how often the data look like for example, in genetics. But here the classical criteria would not work.
Yeah, so what's the problem?
But let's imagine the situation when your N is much, much larger than P. So let's say, like, this is the lone data set when your N is, let's say, millions and P is only 20, or P is only 1,000. N is million, P is only 1,000, then BIC would work perfectly well. And Akaike wouldn't be so bad. Okay, so Akaike would be bad, but BIC would not be bad. So BIC has a penalty which depends on the sample size. So when the sample size is very large, you really almost do not include false discoveries when P is small relative to N. So what I want to tell you again here is that when you work with large databases, you have to be very careful in identifying the setup or the situation into which you are. Okay, is your P much larger than N? Is your N much larger than P? What is your expectation concerning the sparsity? If you expect sparsity, which means that there are not many non-zero betas, you use this criterion. If you expect that many of your betas are different from zero, then you might use some of the regularization techniques like ridge regression. So there are many things to learn and to realize that there is not a single method which would be the best in these situations. Okay. So like in case when P is much larger than N, the Dakaika is very bad, and also BIC is not good because you have this linear dependency of the expected number of false discoveries on P. And this RIC criterion kills this. RIC criterion really kills false discoveries. And what we actually have is a combination of these two. Okay, so this is a modified BIC. And just tell you that we are pretty successful. So I published many articles, and some of them they appeared in the genetic journals. So just to tell you that linear regression is just one situation. You can try to do other models, like here we have zero inflated generalized Poisson regression. So maybe I don't have much time left, so I'm not going really to cover in detail any other topics. Maybe I'll just say to you a few more words. Probably you know logistic regression, do you? Have you heard about logistic regression or not?
Yes.
Yeah, so logistic regression is when your Ys are binary. Okay, so you have binary Ys and you build, use a logit function to predict probability that your Y is 0 or 1. You can also build Poisson regression. So in case when your Y variable has a Poisson distribution, Poisson regression basically models your log of expected value as log of mu is equal to x times beta. Like, so this is Poisson regression. And in our package, we have implemented both logistic regression and Poisson regression. And here you have like position number 5 is the zero inflated generalized Poisson regression. So if you are interested, if you learn about it, so Poisson regression is a very nice model, but very not useful at all. Okay, it's not useful because Poisson distribution is a very stiff distribution. It has just one parameter, lambda, which is equal to the expected value and variance. And it is very rare in practical situations that you have a data for which expected value is the same as variance. So generalized Poisson distribution is a distribution which has one additional parameter which allows to model over or under dispersion. So this is a generalizing Poisson in to a situation when you can have different mu and different variance, different expected value and different variance. And additionally, in practical applications, so we use Poisson regression to model things which are countable. And in this specific article, we use it to model the number of, I don't remember the English name. We call it in Polish stones in kidney. So you have these formations which people develop. So we counted a number of things which appear when you are sick, and many people are healthy. And when people are healthy, this number is equal to 0. So when you look at the real population, this is not really even generalized Poisson regression. This distribution has additional mass point at 0. So this is called zero inflated generalized Poisson. In this situation, we also could use a modified BIC and apply it and have nice results. So this is what we published in statistical applications. So this is a series of articles. Okay, so again, let me just maybe comment one by one. So maybe number 2. So linear regression is a situation when you model residual sum of squares. And residual sum of squares is very sensitive to outliers. So when you have outliers in your data, they are far away from your line. When they make your residual sum of squares very large, and the regression line has a tendency to get attracted by outliers. So you can get estimators which are very bad. And I don't know, have you ever heard about robust regression or not? You have heard? No? So not all of you have heard. Okay, so let me just show you maybe a plot.
Honestly, I don't remember what is that, but we covered that somehow.
Okay, so basically, like in the regular regression, you aim at minimizing. I lost my pen. I don't think I'm. Oh yes, here is my pen. You see my screen? You see my whiteboard?
Yes.
Yes.
So we aim to minimizing sum of yi minus xi beta squared. Okay, this is what you want to minimize when you do regression. So let's call ei is a residual, xi beta. So you minimize sum of squares of residuals, and this is the square function. Okay. Let me just draw a square function. Try to do it well. Okay, so the square function increases a lot when residual gets large. And now when your residual is large because you have an outlier, it has a huge influence on the sum of squares of residuals and has a tendency to attract the regression line to itself, and then the estimates behave very badly. So linear regression is pretty robust towards deviation from the normality assumption by the central limit theorem. You can handle errors which have exponential distribution even. But when they have heavy tails, when we have outliers, when you have a problem. So what you can do, well, you can use a different function. You don't have to use a square function. So you can use a function this way, that initially it is like a square function, but then at some point it becomes linear. So this is what we call this is a Hogbert function. So instead of, let's call it maybe h, so instead of minimizing sum of squares of residuals, we minimize sum of squares of a Hogbert function. And then this large residual will have much smaller influence than in case when I did the square regression. So my function, my function which I add to minimize is not a quadratic function, but the function for which large observations have a smaller influence. So this is a Hogbert function when we replace square function with a linear at some point. And another thing is bi-square function. Sometimes we do it this way. So we do a function which looks this way. So large residuals cannot have a larger influence than this value. So this is what we call an M estimation. Robust estimation. So instead of minimizing sum of squares or residuals, you minimize something like this. Then residuals have much smaller influence, and you can handle outliers in your data. Okay, and then you can also sorry, I'm just trying to find out my slides.
There are theorems that guarantee that the optimization procedure speeds up, right?
One more thing, sorry.
And there are theorems that guarantee that optimization procedure speeds up with these different functions, right?
What do you mean speed up? But it converges.
Faster, right?
Oh, it's not. I don't think so. So, like in case, so you can actually now there are better and better algorithms, but you see, like for linear regression, you have very simple formulas which are analytic, and for M estimates, usually you have to solve an optimization problem. So we had a problem. So like in our application, M estimates actually were much slower than least squares. Okay.
Okay.
So there are proofs about them. They are very nice results, and I think maybe now. So you see, our article was in 2007. I have not been working on current implementation, but M estimates usually are not so quick, because like in case of a least square regression, you have closed form formulas. For M estimators, you really have to solve the optimization problem. So you really find the minimum using some optimization tools like gradient descent, this type of stuff. Okay.
And as you don't have this strict convexity, it would not converge fast. Yeah, Professor, I have a question. Why do we need this? Okay, why not absolute value? Just
why not? Sorry.
Why not absolute value? So you use a Hogbert function or not?
Yes. So this is called L1 regression. So why don't we use L1 regression? Well, people sometimes use L1. So there are some problems related to the fact that it is not differentiable at 0. Okay, so people try to make this function to be smooth. So try to make this function so they are differentiable. But there are implementations when you use simply the absolute value and it is still good. I think it is more problematic from the theoretical point of view. So if you want to prove some results, it's good to have differentiability at 0. But from a practical perspective, people are using absolute value. So this is called, I think, LAT regression, least absolute deviation regression. If you use absolute value, instead of minimizing sum of squares of residuals, you minimize some absolute values of residuals. And this is applied. So you can look for this. It is called LAT, least absolute deviation regression. Okay. And another thing which we did is a rank regression. Rank regression is a situation, let's say that you don't want to estimate your betas, you only want to find out which variables are important. Then instead of working with original y's, you can rank them. And basically, so the largest y has rank 1, the second largest has rank 2. And then you just feed the regression model using ranks instead of original values. So this is a non-parametric procedure. I don't know if you have heard about Wilcoxon test. Have you heard of this or not? Okay, so maybe so I just one word, and then I'm going to slowly close this. One word about the Wilcoxon test.
Yes.
Okay.
Please tell us.
Sorry.
Please tell us.
Okay, I will try. I'm not sure. You see, like I have to go back to my whiteboard. You see my whiteboard?
Not yet.
You don't see my whiteboard. Okay, so I have to stop sharing and start sharing again. Show me on my whiteboard. So the Wilcoxon test is a test where so you have like a regular one sample problem. So you have x11, x1n,
excuse me. It's your first drawing now.
One more time.
We see your first drawing now about the variance. You remember this?
Okay, still okay. So let me just try to share again and do it properly. Now?
Oh, it's a whiteboard. Yes.
Okay.
Yes, all good.
Okay, so we have x1, x and 1, and you have y1, y1, 2. And you want to compare the distribution of x of this IID and this IID. And let's say it might happen that these x's come from and y's come from a distribution which does not have expected value. You know that not every distribution has expected value. You know this or not? Expected value.
Yeah, can you give an example? I just
a Cauchy distribution. Cauchy. Have you heard of Cauchy?
Yeah.
You have heard of Cauchy distribution. This is the density is like the density of Cauchy. I have a problem.
1 divided by 1 plus x squared x squared. Yeah.
Exactly. So I have now problem with my whiteboard, so I will not show you much more, I guess.
Yeah.
I don't know. So I have a problem with everything right now. Nothing is working. Okay, so I don't know, something disconnected and
something freezed.
Something freezed, yes.
Yeah, it's freeze, but at least we hear you.
Okay, so with this, okay, so let me just tell you again. So if you have a Cauchy distribution, then expected value does not exist. So the Cauchy distribution, what does it mean? it means that if you calculate the integral of the absolute value of x with respect to this distribution, it is going to be infinite. So the implication of this is that the law of large number does not hold. So the Cauchy distribution looks a bit like normal, but has the median equal to 0. So it is around 0. But if you average many observations from Cauchy distribution, it will not converge to 0. So the theorem is that if you average 50, let's say, observations from Cauchy, it will still have a Cauchy distribution. So it does not converge. Definitely, central limit theorem does not work for this. So this is a situation when you cannot use a t-test. So let's say that your x's and your y's have a Cauchy distribution, and you want to compare the median, because they have a median. They don't have an expected value, they have a median. So what you do, you can do a Wilcoxon test. And Wilcoxon test will work this way, that you basically put x's and y's together, rank them. So you give x1 rank, the smallest value rank 1, second smallest rank 2, and so on. Then you separate them again, and basically use a t-test, but on ranks, not on x's. And then it turns out that it works very well. It is efficient, and you can actually use this test for testing that the two distributions are the same. And you can use the same idea in regression. Okay, so I'm sorry, I'm just completely frozen with all my things. So we need to converge to the end. So let me just tell you, okay, so that's the point where we have to finish. I did a lot of work on all of this. So I just told you a little bit about the things that you could see, like the references to my work were from 2010. I'm now working